So this was going to be just chatting about. So I'll talk, but just interrupt. And as I say, about what we're going to do. So this is a sort of fundamental assumptions of Mendelian randomization. Bit of sort of history. So it's 21 years. That's the sort of first long exposition of Mendelian randomization. So I was asked to talk in various places in 20 years. So I give more. There's a bit more of a background. Thank you. Perfect. Thank you very much. And then about halfway through, can you bring a beer? Keep me going. So I edited with Shah Ibrahim, edited the International Journal of Epidemiology since 2000, and our first editorial was called Epidemiology Is It Time to Call It a Day? And this rehearsed all the things that I talked about on Friday and earlier about, the randomized trials, not confirming what had been seen in observational studies, etc. Hence the rather grim, hence the title. This is the first time we used. No, it wasn't the first time. But anyway, as always, that's around that end of the 20th century, beginning of the 21st. We always use this cartoon. Has anyone not seen this cartoon? So, you've not seen it. One person has not seen it. Okay, which is it's the according to the New England Journal of Panic-Inducing Gobbledygook, the newsreader reads out, presses the button, and the three things spin round. And today's news story is that coffee increases depression in twins. They then get, that's the news story. And then they press the button, and it just got the next day. It's it comes to the next next combination. Stress causes glaucoma in 7 out of 10 women or something. But every one of those combinations is non-causal, except red wine does cause sexual dysfunction in men aged 25 to 40. So it's a lot of distant memory for most of them. So that was the thing. There was a lot of worry about epidemiology producing very robust replicable findings, hence the sort of triangulation that I talked about on Friday. But so that was very much the motivation. Conventional observational epidemiology founded and reverse causation, not possible to deal with by statistical adjustment, despite what people would often say. And the trick was that you had a genetic variant, which later became called an instrumental variable, which related to your modifiable exposure, and that there would be no confounding because confounders like sleep, socioeconomic position, smoking don't influence the germline variant. And there wouldn't be reverse causation. The early stages of the disease process, atherosclerosis, can't influence the germline genome. So the joint association of the genetic variant with the outcome, basically divided by the genetic variant to the exposure, gave you an estimate of the causal effect. It goes nicely red of the modifiable exposure on the outcome. Now, so this was introduced in the hope of identifying modifiable causes of disease, so identifying things that you could actually change by by manipulating the environment. So then the assumption had to be that there was what called gene environment equivalence, was that irrespective of whether the exposure, say, cholesterol in this case, was influenced by a counterfactual difference in genotype. So, I mean, you could counterfactually have a different genotype. You were just randomly selecting them from your parents. You could have selected another, other genotypes, or now you could CRISPR people, I guess. But that change would the effects of cholesterol on the outcome would be the same if they were influenced by the genetic variation, as if they were influenced by the sorts of approaches that you would like to use for changing the exposure that is a feasible intervention. And of course, in this situation, there's lots of proofs of principle because genetic variant in HMG-CoA reductase, genetic variants in that gene, relate to cholesterol level, LDL cholesterol level, and the drug HMG-CoA reductase inhibition obviously works by inhibiting that gene. So you've got it's rather likely that the differences in cholesterol according to that gene would relate in the same way to differences in cholesterol induced by the drug. So that shows that this you have to have biological reasoning about why that would be the case. So there's gene environment equivalence, and there was evidence that was true in the sort of monogenic disease sphere, where you would have condition pellagra, which is now known is caused by long-term niacin deficiency, and the sort of differential diagnosis of pellagra is a rare monogenic genetic condition called Hartnup disease, which results from a genetic change that leads to poor absorption of amino acids, including tryptophan, which is converted to niacin. So there is a sort of a common pathway, and you would think, and the condition looks very similar. And in genetics, this notion in developmental genetics, like Mary-Jo West-Everhart refers to gene environment equivalence and gene environment interchangeability. Doesn't matter whether it's the environment influencing the gene expression or there's a genetic change influencing the gene expression, the downstream effects are the same. And Zuckerkandl and Willits in a well-known paper, PNAS, said, no doubt all environmental effects can be mimicked by one or several mutations, which I sort of think is not totally true. I don't think that like being hit by a truck, being driven, run over, I don't think there's any genetic variation that could mimic being knocked off your bicycle or whatever. But for endogenous disease processes, which are result of disease through incorporation in biology through embodiment, maybe. So as I mentioned, for cholesterol lowering drugs, was there anyone who wasn't at the triangulation talk? Okay, I'll quickly go for it. Very slowly for you. Yeah. Sorry about it. Yeah, so here you have four matched genes and the drugs that target those genes. So you have HMG-CoA reductase targeted by statins. You have Niemann-Pick C1 protein, which relates to cholesterol transfer across the gut, and that's mimicked by ezetimibe. You have PCSK9 inhibitors, which target the gene PCSK9. You have CETP inhibitors, which target CETP, cholesterol transfer protein. And the point is that the drug trials and the genetic studies match in terms of the sort of dose-response association, the difference in LDL cholesterol and non-HDL cholesterol, which is overlapping, similar. But the genetic studies predict larger effects, which is entirely what you'd expect, because of course with the genetics you have those differences exist from birth, and in Alspac you can show at the age of 4 already, and in cardiovascular young Finns we worked with to show that at very early ages these genetic variants already relate to cholesterol level. So if you have the genetic differences, you have a lifetime difference. So if you group people by their genetic variants, you'll get two groups who have been exposed to different levels of LDL cholesterol circulating in their blood at a group level across the whole of life. Now, you wouldn't expect 5 years of drug treatment, which is what's usually given in trials, to produce the same effect as a lifetime difference in cholesterol. In fact, you get about 35% to 40% of the effect in 5 years. So they scale together, and they're having equivalent effects. It's just that you need to think what the actual estimand is in Mendelian randomization. It's a genetic variant, which usually will produce a difference, or often will produce a difference in exposure, across life, except for some things like, variants related to the nicotine receptor variant I spoke about only produces effects once you start smoking. You become a heavier smoker, or the alcohol-related variants only when you start drinking. So thinking about gene environment equivalence, requires justifying in all situations. I think this is something that just doesn't happen in MR studies, despite Strobe MR says explicitly 0.14 is that you need to justify gene environment equivalence, but no, everyone ticks the checklist saying they've done it, and then when you look, they never have. So it relates to biological processes that are influenced by genetic variation. So if you consider body mass index as a phenotype, you have genetic variants like FGO, which relate to higher calorie intake. So you'd think, you're trying to think about modifying BMI by having differences in calorie intake. That would be equivalent. Genetic variants related to physical activity level, yeah. But then, Miguel Hernan always has given this sort of cute example of, you could saw someone's leg off, and that would reduce your BMI quite substantially, but it would not make you live longer. But of course, there's no genetic variant related to losing a leg. So, so there's obviously, they wouldn't have gene environment equivalence in that situation. And smoking, I put a question mark because I'll come back to it. I mean, obviously, smoking, heavier smoking does lower your BMI, but it wouldn't be equivalent to any intervention you would want to use. So you need to. So I think if you think through this, it really does make you have to be very concrete about what question you're answering. And also, I think it brings you back to realise that when you've got a whole stack of genetic variants related to an exposure, not all of them will be producing the gene environment equivalence you want, because, for example, in a population just of smokers, the CRNA5 variant is indeed quite strongly related to lower BMI. But you're not going to want to use that genetic variant as an instrument for BMI when you're estimating the likely effects of feasible ways of modifying BMI. And that makes you then think, including all these anonymous variants, you do GWAS and you get lots and lots of variants, you don't know what they do. Maybe they're smoking or losing, maybe they're sort of like smoking, and especially as studies get bigger and bigger, of course, genetic variants that relate to getting a disease, genetic variant relates to the disease, and then the disease influences what you think is your exposure. So that's why we brought in Steiger filtering quite some years ago, was because the genetic variant effect or association on the exposure would be less than the genetic variant association with the outcome, if it's the genetic variants influencing exposure through the outcome. So that's why Steiger filtering, works in that situation. A pin-up example was C-reactive protein, because C-reactive protein was found in a whole stack of observational studies to relate to higher risk of coronary heart disease. And if you adjusted for everything, it's still related to a robust difference, attenuated by about a third, or a bit less than a third. And pharmaceutical companies were developing drugs to lower C-reactive protein. Now, C-reactive protein relates to everything. So this is the phenotypic data, just showing C-reactive protein relates to every confounder you can think of, every factor you can think of, always in a way that the upstream factor like body mass index puts your CRP up, age puts it up, they're bad for you, smoking puts it up, that's bad for you, being ill puts it up, that's bad for you, etc. So there's just a huge amount of confounding in this setting. But the genetic variant, this is a cis, i.e. the genetic variant is in the CRP gene, promoter region variant. The genetic variant is related to higher C-reactive protein, but not to it is not related to any of these confounders. The same confounders examined against it doesn't demonstrate any meaningful association. So with this variant, it is doing, the Mendelian randomization is doing what it says on the tin. The variant isn't related to the confounding factors. And if you do the Mendelian randomization, there's no evidence, this doesn't provide evidence that C-reactive protein has any causal effect on coronary heart disease, and pharmaceutical companies stopped trying to develop a drug to inhibit C-reactive protein. Sorts of early sensitivity analyses were bidirectional MR. This comes on to Steiger filtering again. Here is the observational association of BMI and CRP. And of course, the observational association is the same whichever direction you put your equation. The direction your equation doesn't change the observational association. Pure observational associations are not directed. That's the observational association with basically it's this large sample size. There's no real confidence intervals around that. It's a very, very well defined tight association. But then the MR effect of FTO used as an instrument for BMI shows that higher BMI does indeed increase your CRP. I mean, at this time, Nick Timpson had a paper in the Lancet 2005 showing that using MR to show that CRP doesn't appear to influence the risk of metabolic dysregulation and by implication type 2 diabetes. And sure enough, this is the observational association of CRP on BMI, and the MR association is flat. This is using the CRP variant as an instrument for CRP. So you can get that clear distinction. So it was a sort of good sensitivity analysis at the time. And of course, a pin-up of MR was that MR studies, there had been earlier ones by Bone Uddersgaard, but got less attention. But this MR study suggested that HDL cholesterol did not protect against myocardial infarction, despite very strong observational data and a whole stack of trials that were set up and had started reporting negative results by this stage. Now, you can do Mendelian randomization without an IV for the exposure of interest. So when we introduced, it wasn't introduced as an IV instrumental variable analysis, and we gave a lot of the examples in the 2003 paper were not, they weren't an IV, because here you're trying to infer whether sheep dip influences human ill health, and sheep dip contains some anticholinergic factors, and you want to know whether this was making farmers sick, and because there was quite a lot of there was a lot of reported illness from farmers using sheep dip, and there's quite a lot of compensation being asked for in terms of the farm workers, not the actual farmer. This guy probably doesn't own the Land Rover that's pictured there. And of course, we have a wealthy farmer with us, and here's me shearing one of Paul's sheep. But I asked him explicitly, have these been dipped? And he said no. So I was happy to go close to the loaf because it hadn't been dipped in anything toxic that might give me chronic fatigue syndrome. But so this was so with organophosphates and ill health in farmers, what you could do was the beautiful paper by Nicola Cherry, short paper, that had used peroxinase that detoxifies the nasty things in sheep dip, and what they found was that that variant did relate to symptoms in farmers who used sheep dip, but not in those who didn't. So that's quite nice. The genetic variant isn't being used as an instrument for being exposed to sheep dip. It's being used as something which actually modifies the effects of sheep dip on the outcome. So it's the interaction that is being looked at. And something we said in everything we wrote at this time about MR was we were pointing out that we weren't interested in genetic variants in terms of like doing genetic screening, etc. That wasn't interesting. What was interesting is that you can assign causal interpretation to the association between sheep dip exposure and symptoms of ill health. So it was a public health. There was public health implications that were important. It was a phenotypic causation. Does sheep dip influence ill health, that you were using the genetics to actually give you better evidence about that association. And I say quite a lot of the early MR was on this, and didn't have an instrument. So here's another MR without an IV. It's looking at the association of smoking and bladder cancer. Observationally, you have an overall. You have there's obviously if you look at the bottom chart. Current smokers have a much higher risk of bladder cancer, independent of genotype, or in both genotypes. But the rapid acetylation, the slow acetylation genotype related to poor clearing of the nasty things in cigarette smoke, and amongst never smokers, the slow acetylation genotype is related to a higher risk of bladder cancer. In the never smokers who weren't exposed to smoking, you saw no effect. The interpretation here is that again, that smoking is actually influencing bladder cancer. The genotype does not relate to smoking, so it's not you can't say it's an instrument for smoking. The genotype is just relates to effect modification of the external exposure, i.e. smoking. And now, of course, one would also say that the advantage of this is you're not stratifying on smoking, you're not stratifying on a collider, because the genotype doesn't relate to smoking. So that's another rather lovely example, lovely thing for these interactions, basically interaction-based estimation or evidence of causal effects is there's no collider, because the exposure relates to the exposure. So the types of MR study now, your exposure can be either summary data or individual data, or your outcome could be individual data or summary data. Summary data is the sort of two-sample Mendelian randomization. And this is the study design that produces what I will refer to as noodles later. A lot of nonsense papers have come from the two-sample MR. Individual data for the outcome and individual data for the exposure, that is one sample Mendelian randomization. The problem is if you define your instrument, if you're getting, say, a GWAS in UK Biobank for your exposure, and you're applying that to an outcome in UK Biobank, you can get overfitting. The problem is you fit the instrument. The instrument is fitted. It fits too well to the exposure and outcome. So there you need to. There's a jackknife approach that C. Fang introduced. Yeah.
Do you think that does that apply for, like, let's say Hunt and the UK Biobank? They're both European ancestry.
No. It's literally doing it in precisely the same sample. That's when you overfit it, because your genetic. You always explain a bit more of the variance than you would explain if you split the sample and then apply it across the samples, because your model just fits everything, and just the chance fluctuations get fitted, as it were. The chance and those chance fluctuations in the association that exist for the exposure will also relate to the chance fluctuations in your outcome. So also known as winner's curse. If you've done them in different populations, then that's not a problem. And in fact, in the different populations, you slightly underfit. So in the two sample situation, that would then be a two sample situation. In the two sample situation, your estimate is attenuated to the null. In the one sample situation, your estimate moves towards the confounded association. But that's why you can use this jackknife approach. Basically splits UK Biobank into 10 groups. GWAS is 90% of them, then applies those GWAS findings to the 10% and gets an effect estimate, does that 10 times, and then meta-analyzes the 10 estimates. So that's and so this on the IEU website, there's the software for doing this jackknife approach for Biobank. Maybe something should be. I don't. You could set it up for Hunt in a similar way. you see now, because of UK Biobank and Hub will be the same, you often see this hybrid design, which is where you take for your exposure, you take this is exactly what you were asking. What you would be doing if you took the biobank exposure GWAS, you would take that and apply it to hunt. That would be this hybrid design, which is common is now very common because of UK Biobank. Hunt will use it, and there you don't. If anything, you would tend to slightly underestimate, but when you simulate the underestimation, it's pretty small. Underestimate the effects, and then I'm not sure I've ever seen using individual data for the exposure and summary data for the outcome. Not really seen that, but so that's the unicorn study design. So where did the name come from? So we were working on Una Fallon was a medical doctor doing an MD she's from Ireland, and where Southern Ireland, the contribution to cardiovascular epidemiology was the identification of homocysteine as a potential coronary heart disease cause. The good news about homocysteine was you could lower it with folate. So there was thought that this would be a mechanism of reducing risk. The thing is, homocysteine was like C-reactive protein. Most bad things put homocysteine up. But we published a paper in Heart where we discussed the measurement error in the confounders, and we argued that actually the residual elevated risk was entirely in line with what you'd expect because of measurement error confounders. But one of the major promoters of folate supplementation in the UK wrote a very cross letter saying that this was untrue. He didn't believe it, and in that we wrote a response to that and said that the way you could actually estimate this would be using the genetic variant in the MTHFR gene, which relates to a difference in homocysteine levels, and that your estimate was rather imprecise. There's an imprecise estimate seen, and therefore the sample size was pretty small. And in the usual over-the-top way, I put the through Mendelian randomization, there's tests essentially that provide association, blah blah blah. Conventional epidemiological studies have a limited future, so I'm banging 2001. I'm just like a stuck record, really. But luckily, I'm heading towards retirement, so I'll won't be going on as a stuck record for too long. But I think I was saying the same thing: randomized trials or further studies utilizing Mendelian randomization. And the reference I gave there to Mendelian randomization was to this paper. And I'd taught the London School of Hygiene when I was teaching epidemiology. I taught a course at the end, probably around 1991 on, I was teaching a course around this time on alternatives to randomized controlled trials. How could you get evidence that worked through randomized controlled trials? And I don't know how I'd come across this, but I'd come across this somewhat obscure paper because it was discussing not doing randomized trials, and I'd searched for, I don't know how I found it, but and it had suggested this amazing design for examining whether bone marrow transplantation reduced the risk of mortality in children with hematological and lymphatic cancers. Now, no parent would allow, or very few parents would really want their children to go into randomized trials because the outcome is pretty grim for those conditions in children. And bone marrow transplantation was just coming along, and it seemed to offer some hope. And what they came up with, they said, it can be assessed without the need for formal trials by taking advantage of Mendelian randomization. But what they were talking about wasn't looking at genetic variants related to an exposure in the way that we now think it, related to biomarkers. Their absolutely ingenious solution was that if you were a child and you needed a bone marrow transplant, the most common successful source of a bone marrow transplant by far was a sibling. And of course, if you had an HLA compatible sibling, you were more likely to receive a transplant because you would have a compatible someone to actually give a transplant, provide you that transplant. So if they had a compatible sibling, you're more likely to get a transplant, and therefore if you can compare the whole group that received or did not receive a transplant on the left and didn't on the right, that is equivalent to an intention to treat trial. They didn't. This is my picture. This is my drawing of their design. You were doing an intention to treat analysis, so the sort of analogy with the randomized trial is rather neat. The only fact is that you should take into account whether the number of siblings they have, because number of siblings relates to risk of acute myeloid leukemia could relate to the outcome, but that was a minor modification that would. But it was really ingenious, and studies were then set up using this approach. But so that's what I'd seen, and I'd remembered that term, Mendelian randomization. So that's why I used it, taken from that paper. You didn't have wonderful online searching facilities in 2001, as you do, 2000 as you do now. And I've since found earlier examples of people proposing the same thing and using different terminology. So in 1989, so a couple of years before, this different group, Jonathan Buckley et al., had written a paper, and they discussed a more rigorous approach. It's based on the biological randomization of availability of matched sibling donor. So if I'd seen this, I would have said biological randomization, because I always like getting the earliest thing. But then also in 1989, you had in an animal situation, you had people discussing biotic randomization, which of course is what it is. The randomization occurs at meiosis, or there was this genotypic randomization. This was in 1989. This was in an evolutionary context. And 1993, calling something, they called something genetic randomization, or prospective genetic. I can't really read it. I can't remember what it said. I think it is prospective genetic randomization. Randomized, a genetically randomized comparison. No, I can't bloody read the thing. Oh yes. Anyway, so we could have been biological randomization, meiotic randomization, Mendelian randomization, genotypic randomization, genetic randomization, reverse genetics, because that's all it is. Andrew Hattersley, when I went to talk to him about this idea, just said, it's just reverse genetics, because very confusingly, forward genetics is when you take an outcome and then you go and find the genes. Why that's called forward genetics, I have no idea, but that is called forward genetics. And reverse genetics is when you have a gene that you know what it does, you think you know what it does, you then look to see what the phenotypic consequences are, and that just is Mendelian randomization, or genetic cause linkers. I might have been less antagonism of it. I think some people didn't like the name Mendelian randomization, but so maybe human reverse genetics or something might have been better. But this is me being rude. I don't care what you call it, but for fuck's sake, read the papers you cite. This is the Graham Wheatley paper has now been cited massively, which is good. It had 19 sites in 2003. It's now had a vast number of sites. And this is Mary Schooling cited it as a study design that used genetically determined differences in exposure to test whether a biomarker affects disease. And there had been decades over which there'd been use of genetic variation of the biomarker to deduce the causal effects of that biomarker on a disease. So call it what you like, but actually read the papers. You can cite it. Anyway, and it was introduced strictly Mendelian principles, i.e. it should be within families. So you were trying to follow the genetic variant across families, because that it's meiosis, which is randomized. And if you have, say, siblings within a family, then it is true randomization. There are randomized genetic differences between siblings. That's what we said was analogous to RCT at the time. We gave this example, this sort of figure of following genotype from mothers and fathers down to the offspring genotype phenotype. And then it said that the problem was there were virtually no family studies of any size. There was only one example we had of within family MR, which I'll come on to later. And therefore, in populations, you could try and deal with population stratification. It was a problem, and thus the Mendelian randomization and genetic association studies is approximate rather than absolute. And I sort of wish now I'd just carried on calling it approximate Mendelian randomization. AMR is what people were doing. Virtually all Mendelian randomization was just approximate Mendelian randomization. And rather bizarrely, in showing how slow on the uptake I am, even though I taught a course in instrumental variables, I didn't recognize this many years before. I didn't recognize that what was being done in some cases in Mendelian randomization was an instrumental variable. And the reason I didn't is because of people like Johan, these economists who used instrumental variables. And when I was teaching this, I was working on childhood diarrheal disease in Nicaragua, and I came across these instrumental variable papers. This is back in late 1980s, early 1990s. I came across these papers which used distance from the water supply as an instrument for amount of water used. And these people had obviously never been in any of these places. They'd taken data, because if you'd been in rural Nicaragua, you'd realize that the places that were distant from a water supply or a pump were very different. There were from places that weren't. And the idea that this was in any way useful. So I filed instrumental variables under in my brain it was filed under bullshit, or higher order bullshit. And so I thought this Mendelian randomization was pretty neat. So I didn't think of it. I therefore didn't think of it in terms of instrumental variables at all. And this was one of the sort of face plant moments when I invited Duncan Thomas, Davey Conti to write a commentary on a reprint of a piece by Martin Catan from 1986, where in a letter in the Lancet he'd said you could use ApoE genotype of genetic cholesterol as a way of finding out whether low cholesterol increased the risk of cancer, because there was a lot of dispute about whether low cholesterol caused cancer. He didn't do a study. It was the reverse causation, and the letter had been cited twice by 2003. It's now a citation. It's now been cited very, very, very frequently. Anyway, they wrote, so they attributed it. They said, particular those with the Ibram shown that the magnitude of the estimated effect, which we had done. I'd calculated these things, which were IV estimates. I'd misspecified the standard errors actually. So you can write in and say that the standard errors in the 2003 paper were misestimated. But they thought and I thought, God, face plant when I was told that it was an instrumental variable analysis. Anyway, instrumental variables. So instrumental variable, the instrument here, Z, has to relate to the exposure of interest, X, and you can test that. That's the one thing you can test. There should be no confounding of the genetic variant, i.e. the instrument, and the outcome. That's the second IV assumption. Now you think, well, how are you getting confounding? How can a confounder influence the genetic variant? You're thinking it's a distributional phenomenon, and population stratification you can think of as a sort of confounder, where the population stratification influences the outcome, because some ancestral populations will have different outcomes than others, independent of any particular genetic difference. And also that influences the distribution of the genetic of the instrument. So that's the form of the confounding there. But the other similar, well, not the other very dissimilar, sorry, form of confounding would be dynastic effects, because one thing which actually does directly influence the genetic variance is your parental genotype, because you are randomly sampling genotypes from your parents, or your parents are randomly assigning them the gametes that become you. So that is plausible. You could get the parental genotype would then have to influence the outcome of exposure. Because the exposure influences the outcome, you actually then have got confounding of the instrument with the outcome if you have that situation. So dynastic effects, though, you'd have to think of mechanisms for them, and one very obvious mechanism of that dynastic effect would be that the maternal genotype would influence your intrauterine environment. So, and this is the one example we had of a family-based MR in the 2003 paper, was with this NTHFR variant. If your mother has a genetic variants that increase homocysteine levels, lower her relative folate activity, then that increases the risk of neural tube defects. And of course, we know through randomized trials that folate supplementation from very early in pregnancy does reduce neural tube defects. Then you've got this beautiful negative control, because the father's genotype doesn't influence the intrauterine environment. So this is just like the negative controls I talked about in the triangulation with the paternal smoking as a negative control for maternal smoking. So you've got beautiful negative control, but of course the fetus inherits 50% from the mother and 50% from the father. So on average, their genotype will be a genotype related to higher homocysteine, lower folate activity. But it would be a mistake to think that it's the offspring genotype, the neural tube defects that's generating the effect. It's just that the offspring genotype reflects the maternal genotype, which is the thing which actually has the influence on the outcome through the dynastic effect of influencing the intrauterine environment. And yet again, like stock record, we just said this doesn't. This shows it doesn't suggest that genetic screening is indicated. Rather, it demonstrates that environmental intervention, e.g., folate supplementation, may benefit the whole population independently of the genotype of individuals receiving the intervention. So this is always not intended to be anything to do with genetic screening. Another dynastic effect. That's the maternal genotype influencing through the intrauterine environment, influencing the outcome and also influencing the exposure. So that's a form of confounding. Or it could be that the parental genotype could influence the environment. So, for example, you have your parental genotype relates to higher educational attainment, there'll be more books in the house. If your parental genotype relates to more smoking, the kid on average will grow up in an environment where there's more smoking. If the genetic variant influences alcohol consumption, they'll grow up in an environment with more alcohol consumption, etc. So these are plausible forms of confounding produced by through dynastic effects, and that's why the within SNP studies were powerful. And then the third assumption is that there is no effect of the genetic variant on the outcome, which is not mediated through the exposure. There's no non-exposure influence, i.e. there's no red line here going through some phenotype, because genes can't influence outcomes except through some phenotype. And I was worried about, and this worried me for a long time. I was worried about the way economists wrote this is that Z is independent of Y conditional on X and C. I thought, well, this is bloody useless, because the whole point of Mendelian randomization is you don't know C. C is the confounder. So why do this? Well, it's because if you just condition on X, of course, you are conditioning on a collider, because your genetic variant influences X. When you adjust for or stratify for X, anything else that relates to X, like if C influences X, will become related. Will indeed become related to your genetic variant. So I'm sure that these days everyone knows about collider bias, but this is my sort of my favorite. example or thought experiment about collider bias is if you imagine a US university, US universities, students can get into them either on a sporting scholarship, they love baseball players and basketball players, etc., or an academic scholarship. So, and if you've got one or the other, you'll get into the college. So let's imagine that in the population, the dashed line, in the whole overall population, there's no association at all between sporting prowess and academic ability. The correlation is zero, no association at all. But if you went on to a college campus, one of these colleges that has these admission policies, you went onto the campus and there's a sort of student over there, and you throw a ball at the student, and they try and catch the ball, and they fall over backwards, and it bounces off their head, so they land on their back. You can go up to them and say, "You are very clever," because there's no way they've got admitted to college on a sporting scholarship. So they must be admitted on an academic scholarship. And in fact, in this situation, the two things become negatively correlated by about 0.6 or over 0.6, just in the people admitted to college. So that's collider bias, and that's why you don't want to condition, you don't want to adjust for X. There's lots of papers. There's a sort of letter in the JAMA, one of the JAMA journals that Marcus Manappa and I wrote a cross letter about, which claimed to have developed a test for exclusion restriction, which was adjusting for the phenotype, adjust for the exposure, and this would, and of course, if everything's mediated through the exposure, it should go down to null. And of course, there's measurement error in the exposure, but it's also collider bias. And I think there's been 100 years of incidental variables in economics, and we all know that economists are geniuses, and no one's thought of a test. There's no test for the exclusion restriction. Now, if you think you've come up with a test for the exclusion restriction, either you should get a Nobel Prize, or you're wrong. And so far, everyone who's come up with a test, claiming they've invented a test for exclusion restriction, have been wrong, being Nobel Prize worthy. So that's horizontal pleiotropy.
Just a side question: do you think that goes also for this kind of a popularized mediation analysis from Van der Weve and so on, that it's the same, that you can say that either they are genius or they're wrong, and they're most likely wrong when they're doing mediation analysis?
I absolutely think that, yeah.
Yes.
I think it's a con, yeah.
Yeah.
It's amazing.
Why does someone say so?
I've said it, 650-page book.
Yeah.
He's a very smart guy, but it's wrong. Yeah. Anyway, that's my view, yeah.
So you should
The only
we should stop doing it.
the only way you can do mediation analysis, of course, as I'll talk about, is with Mendelian randomization, because you're instrumenting it. So, yeah. So you estimate you don't adjust for the exposure, you just estimate the effect that you'll get through the exposure and see if it fits with what you get. So you can do mediation analysis, but only with Mendelian randomization. Well, I think you'll try it in some trial situations. But yeah, but just in observational data, it's I do think it's wrong. So that's horizontal pleiotropy. Remember, it must go through an exposure, because a gene can't influence an outcome except through an exposure. There's also vertical pleiotropy. So horizontal pleiotropy, you can see why it's horizontal, because it's not through the, it doesn't go through the there's an effect that's not through the exposure. There's also vertical pleiotropy, where a genetic variant influences one exposure, one phenotype, and that phenotype influences a downstream phenotype. Now that form of pleiotropy is not a problem for Mendelian randomization, as long as you know which is the most proximal phenotype to the genotype. In fact, as you can see from the diagram, Mendelian randomization just is exploitation of vertical pleiotropy. So maybe that's another name. Maybe I could have just called it vertical pleiotropy or something. harnessing vertical plyometry. I should write that down. Anyway, and so when you see papers that say, there's a vast amount of pleiotropy across the genome, it's true, but a large chunk of that pleiotropy is vertical pleiotropy. Because of course, a genotype like FTO that influences BMI influences so many things. That genetic variant relates to coronary heart disease, to some cancers, to needing a larger belt, to many things are influenced by BMI. So a lot of the pleiotropy may be vertical across the genome. But now, and then you can have sort of mediated vertical pleiotropy where the genetic variant influences something upstream of your exposure. Now, if the upstream phenotype doesn't influence your outcome, then that's fine. That's all okay. But if your upstream phenotype also influences the outcome, then you might misspecify the primary phenotype. So imagine something like FTO influences BMI and BMI influences CRP, as I showed earlier. If you took FTO to be an instrument for CRP, you would obviously get the wrong answer because BMI influences your outcome. Now, if you know what the upstream factor is, then you can do Steiger filtering. That allows you to identify the primary phenotype because genetic variant would explain more of the variance in BMI than it would in coronary heart disease or CRP. So you would know that genetic variant shouldn't be used as an instrument for CRP because it has a stronger effect on BMI. So that's what Steiger filtering does. That and helps you orientate to know whether you have misspecified the primary phenotype. But that setting where there's sort of an unknown phenotype, just a phenotype you don't know that influences both your exposure and your outcome, is probably very common, and that causes the biggest headache for Mendelian randomization. Yeah.
Sorry, are you taking questions here?
Yeah.
I'm just curious about BMI as a primary phenotype.
Yeah.
How certain are you that BMI is a primary phenotype?
No, that's a very good
underlying
point.
phenotypes that, I don't know if the Steiger filtering would identify phenotypes that aren't kind of defined.
Yeah. So I suppose what I should have said is more primary phenotype because FTO you can show in Alspach, for example, Nick Timpson showed that the FTO variant relates to higher calorie intake. So that then you have the higher calorie intake relates to higher BMI. So if you actually had the calorie intake data, then you'd want to Steiger filter with that. So that's one of the real issues for most genetic variants. We don't really know what the primary phenotype is. But BMI is more of a primary phenotype than CRP, if you see what I mean. So it's BMI is CRP is downstream of BMI, and Mendelian randomization shows that. So that's a very good point. So interpreting the IV estimates requires all of the IV assumptions. I've already discussed this. The exposure period is often not well defined, because the genetic variants are influencing things across life. It may relate only to an exposure during a critical period. An example there is that Mendelian randomization demonstrates that vitamin D reduces the risk of multiple sclerosis, but it might well be that it's only your level of vitamin D when you get infected with EBV. That's the important thing. So if when you're infected by EBV, your vitamin D is higher, then that leads to a more favourable sort of immunological response, which reduces your risk of MS, multiple sclerosis. But of course, because your genetic variant influences vitamin D right the way across life, you don't understand that it's just that critical. It's that critical period. Giving people vitamin D after they've been infected might well be not useful. So it's and these exposure periods might be very early on, and they could even be fetal. But like, you know, one of the arguments for why base inhibition, which was something to try and reduce Alzheimer's disease, and it's one of the genetic variants found early on related to Alzheimer's disease, and it could be druggable. So Merck developed a drug, didn't reduce Alzheimer's disease. It could be that the effects are happening, you know, very early in development, and so giving people things at age 50 isn't going to work. I'll skip that. And for categorical exposures like a disease, having a disease, your MR can't, your usual MR, you know, be it 2 sample or 1 sample without timed events, can't tell you whether it's actually the actual disease occurring or it's the genetic liability to the disease which influences the outcome, because, you know, because often rather few people in your population will have actually developed the disease. So if you think you're doing MR of prostate cancer on outcomes, then you're actually looking at the genetic liability to prostate cancer, unless you have timed events. I have not seen any paper that's ever done this. You could, in principle, do an MR model if you knew when people's prostate cancer occurred, but I've never seen a study doing that. And the fact that prostate cancer's example is quite a nice example, because you can show its genetic liability, because some of the things that are apparently caused by prostate cancer, if you do MR, 2 sample MR of prostate cancer, they also show effects in women who don't have prostates. So it can't be, obviously, it can't be prostate cancer itself, it must be the genetic liability. So for some categorical exposures, you have to have a good argumentation about why you think it's actually through the exposure. You're not just looking at genetic liability. And in terms of, you know, within medicine, a major problem is it generally relates to disease occurrence, not outcome. So, you know, virtually all the GWASs are having a disease, like having lung cancer. And of course, you do the GWAS of lung cancer, the top variant is related to smoking, as I discussed earlier. But I mean, stopping smoking isn't exactly a great treatment for lung cancer. You're still going to have very high mortality once you develop lung cancer. But it does. So MR actually tells people about things that cause disease, not about treatment. And that's one of the sort of things that I think with the whole genetics revolution was sort of missed. People somehow thought GWASing disease was going to tell you about treatments. I mean, it's not. I mean, and that's why I was so interested in genetics. It doesn't tell you about treatment, it tells you about prevention. But many people want to know about treatment, and you would ideally want to use Mendelian randomization to look at treatments. Now, there's lots of sensitivity analyses through them. So people know, and Alan Sanderson's review reviews the sensitivity analyses. I'm happy to discuss them after. I think one of the nicest sensitivity analysis is using negative controls or 0 relevance points. And an example of that is with alcohol intake, the risk of disease. I don't know if in Norway, when these papers appear saying that alcohol protects against something, do they get in the news? Are they popular? So this was a study from a few years ago in the BMJ that suggested that alcohol intake was related to the lower risk of a whole stack of cardiovascular diseases. The papers loved it. This was the Guardian. Moderate drinking can lower risk of heart attacks. A study going down market to the Independent, drinking a pint of beer a day linked to reduced risk of heart attack. Mail Online, these are just lower and lower level newspapers. Shears, drinkers who have 1 glass of wine a night are at less risk of heart failure than teetotalers. Daily Star, really low grade paper, drinking alcohol slashes risk of heart problems if you drink this much per week. Brilliant, thank you.
With that Norwegian study,
Yeah.
found that teetotalers had lower risk of hip fractures.
Yeah. Well, that might be close.
Don't drink, you are at increased risk of you had an increased risk of hip fracture if you were a non-drinker.
If you're a non-drinker.
Yeah, when you were a non-drinker.
Yeah. Even though you were more likely to fall
The best
over
was to drink moderate.
to keep your hips intact. And this is time from the US. Alcohol is good for your heart most of the time, rather sober sort of comment. And my favourite was the Irish Times. Moderate drinking may cut risk of heart disease. So I was like, that's okay. But then down below it says, moderate drinking may be good for your heart, and even heavy drinking may lower your risk of heart attack. A new study suggests, indicates. No. So you can study alcohol through Mendelian randomization, especially in East Asian populations where a high reasonable proportion of the population carry either 1 or 2 copies of a knockout of the acetaldehyde dehydrogenase variant. And that gene that produces an enzyme which metabolizes acetaldehyde to acetic acid, which is then excreted. And in cartoon form, when you drink ethanol, it puts up your acetaldehyde, and acetaldehyde gives you what the sort of negative effects of drinking. So you get a headache, you get sort of palpitations, you get flushing, you get guilt. Actually, no, not guilt, but all the other nasty side effects of drinking are generated by acetaldehyde. So you want to have your acetaldehyde cleared. And the people who are homozygous for ALDH2 knockout just don't drink. This is a very old meta-analysis we did in studies of men and women. And amongst the men, you see that the heterozygote, no one is drinking at the homozygous non-variant, but for the men, they drink generally around intermediate level. If they are heterozygous and they're homozygous wild type, they drink the most. But in women in these populations, when these studies were done, the women weren't drinking. Didn't matter what their genotype, they weren't drinking at any genotype. So that gives you a lovely zero relevance point, because amongst women, the genetic variant doesn't relate to alcohol. But if there's pleiotropic effects, you'll see the pleiotropic effects in the women. Whereas amongst men, it influences alcohol, but would also have the pleiotropic effects. So when looking at blood pressure as an outcome, you see that in women, there's no relationship between the genetic variant and blood pressure. Amongst men, there's a dismayingly large effect, such that if you are homozygous for the null variant, you have considerably lower blood pressure. So this shows that alcohol does indeed put up blood pressure, which has also been shown in randomized controlled trials. So you should take an ACE inhibitor with your beer, because it definitely has adverse effects to alcohol. And I'll skip that. And in
I just wonder if you put this into this Jeffrey Rose perspective. This goes for those who the population effect would be to those moderate drinkers. We who drink much more, we would not contribute much to the population health effect. So we could continue drinking, but those moderate drinkers, so they should drink less. That's
I know exactly. Believe us, yeah. So lost cause, yeah. You could reduce, get rid of 90% of the risk by making the moderate drinkers drink a half pint rather than a pint. Exactly. So these are data from China, where you have it's rather a lovely study, the Kadoorie study, because you have women who don't drink in 9 out of the 10 centres. They drink a little in Shanghai. The men who drink a lot in some of the centres, but in some of the centres, the men, especially where there's some centres in Muslim areas, the men are hardly drinking. So you can get these groups defined by chromosomal sex and region and where they live, that drink very different average amounts, and you can do the Mendelian randomization in each of those groups. So you can get a dose response effect, and you can look at the non-linear effects without stratifying by a collider or using a method which produces impossible findings.
So just a question of, I think that's a really nice study, but all these regions really in a way exchangeable.
Well, so the point is, it's not whether it's changed, it's whether they're a collider. So is it really true that your genetic variant through the alcohol is likely to lead to people moving. I mean,
No, but these regions could have a lot of other factors.
No, absolutely, but no, but the analyses are entirely based on your genetic variant association. In these, it's a Mendelian randomization. It's entirely a genetic Mendelian randomization within each of these 20 regions.
But it's a combination of region and genetics.
And sex. It says that, but what it does is that actually, if you read the method, it does the MR within the genetic effect is through an MR effect within these regions that have different levels of alcohol consumption. And it's a method that should be extended, because if you can get places which really do have different levels of exposure. But the problem is, it's very rare that you get such great differences in exposure by non-colliders. The nice thing is you're not stratifying on a collider. And what you see is for stroke, you see the U-shape, the U-shaped curve. The moderate drinkers are doing best, but then when you do the MR, you sadly see it's just bad. So ACE inhibitor. When I was in Glasgow, I bought this beer. I remember taking this photo. I thought this was fantastic. A beer called Old Mortality. Just brilliant. And it's so there's no way I can't sue this company and say, like people sue tobacco companies. I can't sue them and say, come on, I've had a stroke now, and that's because of your beer, because they call the fucking stuff old mortality. I mean, that's better than a health warning, isn't it, on the beer? Also, I showed that again. I showed all these set-through analyses. So the template for an MR methods paper, so if anyone wants to do an MR methods paper, is your introduction is current methods have limitations. You then your method section, we simulate data and apply our new method and a handful of previous methods to these results. Our method performs best. And conclusions are we are the champions. That's every MR methods paper summarized. Okay, what it is, I'm going to skip this up and go on too long. The bottom line of all of this, it's getting close to bar opening time. The bottom line of this is that when you've got you measure something like blood protein, and you GWAS the blood protein, and you get a genetic variant, maybe a cis variant that relates to the blood protein, and you think you're instrumenting blood protein, but in many cases. The blood protein is not going to be the causal factor. So this is sclerostin, which is I'll get back to our lights. Sorry, cutting down on drinking time. But there's Mendelian diseases where people have sclerostin is knocked out. They have extremely high bone mineral density, and a drug that's developed, monoclonal antibody, was developed entirely because of the evidence from genetic studies. It's an exemplar of genetically introduced. A genetically derived drug. So you look at, you have circulating sclerostin, you GWAS circulating sclerostin, you get a couple of genetic variants that relate to it. Neither of these are actually cis variants when we did this. And your MR does show that an increase in circulating sclerostin level relates to lower bone mineral density and higher fracture risk. So the MR is sort of getting what looks like a nice answer. I'll skip that. So then, the follow-up GWAS with much larger numbers, we then got a cis variant in SOS. And then what the variant we had before, the top variant before, was the B4-3, which there's a mechanistic. There's arguments about why it does relate to sclerostin and RIN3, but two variants that relate to circulating sclerostin. But there's lots of other variants, and the problem is that for a lot of the variants, it appears that the variant influences bone mineral density. And then if you've got higher bone mineral density, you upregulate sclerostin, which lowers your bone mineral density, because biology is a homeostatic system. So in many systems, you will have this sort of homeostasis. So increased bone mineral density leads to increased sclerostin levels, decreased sclerostin levels leads to increased bone mineral density. So you have this sort of circular thing. So you obviously don't want to include the variants that relate to high bone mineral density and higher sclerostin. You get exactly the wrong. You get not just the wrong answer. You get quite a strong answer in the wrong direction. So you Steiger filter them to remove the ones. You Steiger filter everything against bone mineral density, and you end up with 4 that go in the right direction. But the point of what I was saying really is that the SOST effect, that SOST variant effect on bone mineral density is way bigger. The IV effect, IV estimate is way bigger than the IV estimate for the others, and for fractures it's the biggest, and it's rather the precision is less. But the reason for this is of course, well, I think that the obvious reason for this is that circulating sclerostin in your blood does not influence your bone mineral density. And when you give people the monoclonal antibody against sclerostin, it goes to their osteocytes and it stops sclerostin being manufactured in the osteocytes. It's the sclerostin, local sclerostin levels in your bone produced by the osteocytes that matters. And the SOST variant, which is actually the SOST gene, will be producing larger local differences. The things which influence your circulating sclerostin, it's just the stuff that leaks out of your bone, and the B gallant variant might well be influencing it through reducing excretion levels, for example. So really, what you want to instrument is sclerostin activity in bone. But we always just take these things in blood and GWAS them and think that we're getting the right phenotype. It's probably quite rarely is it the right phenotype. It is the right phenotype for cholesterol and coronary heart disease, which is why that is the pin-up of Mendelian randomization, because it's your circulating cholesterol that leads to atherosclerosis. But for most things, a lot of disease, tissue diseases, that's not the right. It's not the right phenotype. I put too much in there. Sorry. I talked about this in the triangulation talk, so I will skip this. It's just showing that it's bone mineral, childhood body mass has an effect on reduces the risk of breast cancer. There's lots of proof of principle studies. In fact, I talked about. I talked about this earlier, didn't I? Were people here? Was everyone here for my earlier talk? Everyone but one person. I won't go through it, but yeah, everyone's there. So, and this is the when I said on the next PowerPoint, because the next PowerPoint, I think this is rather beautiful with all the different all the different adult onset phenotypes do relate more strongly to adulthood BMI, except severe autoimmune type 2 diabetes, which will include this sort of type 1 diabetes of adulthood onset, and LADA, which is driven by childhood body size, even though you're now looking at a disease onset when its usual onset stage 40, 30, 40, 50. Childhood body mass, it's a childhood BMI which is increasing the risk of it. So some process is set in place, which is generating the autoimmunity is set in place in early life, influenced by your rapid childhood growth. I showed these as well. Yeah, I showed these. Okay, so this is how is it really? Chronicle of a Death Foretold. I'm sure some people have read the wonderful Gabriel Garcia Marquez on the Death Foretold. Appropriately, was from someone from Latin America, Fernanda Hartwig from Brazil, who wrote a paper in 2016 on two-sample Mendelian randomization, the problems with two-sample Mendelian randomization. And he wrote this paper when this paper was written just when he in 2016, difficult to imagine, but there was only a relatively 150, 200 real actual empirical MR papers. It was quite rare until the two-sample revolution. And he was writing at a time when the two-sample had just overtaken one-sample MR. Now 95% is two-sample MR. and demonstrated how a paper looking at C-reactive protein schizophrenia, how they'd miscoded the genetic variants and other problems, and that led to this is the first MR retraction. We got scientific reports to retract the paper, but based on Fernando's paper saying, look, this 2-sample MR is starting to produce sort of nonsense studies. It's just too easy to do. Chip Hamani set up a whole system which does MR of everything versus everything. And he sent to Nature Genetics, and in 2000 we got really lovely, positive, short reviews back, and never revised it. It's never been published, but it is available within Epigraph DB. So you can look up any 2-sample MR answer you want. You go to the Epigraph DB database, and then this is the takeoff of. Bjorn Asloff's going to get me a beer. That's very kind of you. This is the ridiculous takeoff of MR papers, almost entirely due to see when Fernando was writing, it was 2015, 2016. There weren't many MR papers, and it is a literal exponential. So you can show that in 16.5 months, every single scientific paper will be Mendelian randomization paper. So that's where it's going. So, and a lot of them are just nonsense. They're doing 2-sample MR on things which you simply cannot instrument, like GWASing noodle intake. So you have some sort of specific variants that relate to people liking noodles. Not likely. Causal effects of COVID-19 on cancer risk. This is fantastic because the outcome cancer data were all collected before 2020, so not a single person had COVID. This is the interpreting as genetic liability. This is why you should interpret things as genetic liability. And Wiley are predatory publisher. Lipid-lowering therapy on multi-cancer prevention. You GWAS whether people are on simvastatin or atorvastatin. Atorvastatin is you've got a better GP, so you're probably better off. I mean, you don't get a genetic variant which actually tells whether you take it, which sort of statin you take. You don't give GWAS what drugs people are taking. Particulate pollution from UK Biobank, that is inferred from your postcode. So you are GWASing someone's postcode and then using that. And so it only tells you about air pollution postcode. You think we're getting population stratification. Or habitual consumption of alcohol with meals of lung cancer. What do you reckon? Is drinking alcohol with meals good for you? Does it prevent your lung cancer? As opposed to drinking alcohol in the Three Lions, where you can. I like the fact you can still smoke outside bars in Norway. It's one of those attractive things. But yeah, as you imagine, you're drinking, you're GWASing something which is related to health protective behaviour, general factors. You just can't do it. And this is Mendelian randomization package, video, and Mendel code pack, catalog, and price. This is translated by Si Fang from the Chinese. This is on Chinese websites. We're a medical research team composed of a number of 985/211. Well, what that means is there's a set of universities in China which are the top universities, and they're called either 211 because there's a 211 which are the. So they're saying we're doctors from those, and we've been deeply engaged in Mendelian randomization analysis. Published a high level, XCI stands for scientific citations, Science Citation Index, because you get paid in China. If you publish a paper at a university, you get paid according to the journal has to be in the Science Citation Index, and then you get paid by the impact factor of the journal. You get a bonus, and they've developed a package. And they've got 15.3 points Mendelian randomization article accepted. So what they're saying is they've got a paper in a journal with impact factor 15.3 is pretty good. This also means you can look it up. So I decided not to put it up when I did this. I know what paper this is. And what this is saying is we did this paper. This first. These are paper factories. They take you through, they help you, they do the analysis. They help with the writing of the paper, and there's a simple I love this, course and code package an exclusive teacher to send one full text. The text guide price is 339 which is about 250 quid, I think. And then you can go for a package for 5 papers and you get a discount if you're going to pay for 5 papers. The student said, "I'm not proficient at getting started just now. In the future, it will be done in minutes." You can do this in minutes. And anyway, and then you see these there's all these completely different author groups. There'll be just a space within a few days. These papers will appear all on the same topic. There's the microbiome and erectile dysfunction. All these erectile dysfunction. These are all different groups. Yeah, it's the same thing. They're publishing the same fucking thing, but and these are, you know, the Wiley journals. These are quite high impact factor journals. It's amazing that they all have the same idea at exactly the same time. And there's 22 of them. Oh, microbiome and you've got the microbiome. Come on, you can compete here. Have you got erectile dysfunction? I wasn't asking you personally. Only after we're supposed to say only after too much red wine. Is the answer. No nulls. So the MR when MR was good. The major things, you know, the useful things MR did was to show how many things that drug companies wanted to target, like C-reactive protein, like fibrinogen, like HDL cholesterol, were not. But there's now no nulls. None of these papers are null. The microbiome causes everything, including etc. So there's no nulls. And then the third thing is it's just a load of methods that no one understands. I call it numbskulls because they're difficult. They make your head hurt trying to understand the methods. His vitamin D in human health is brilliant. I talked about this on the triangulation. There was a few people not at the triangulation. Who was not at the triangulation? 2, 3. I'll run it. So basically, you got vitamin D is null for everything but multiple sclerosis. Is basically like cardiovascular disease, cancer. That agrees with all this vast number of randomized trials. They're null on cardiovascular disease, all different forms of cancers, etc. And bone mineral density fractures, null for that, null for everything. Essentially, the MR studies agreed, but there had been these papers and you couldn't get these people stopped publishing, you know, big journals stopped publishing these papers. But then there was a study claiming to be able to look at a dose response effect, and these are how the curves match up. This is all cause mortality observationally. And you use this method, which is the residual method, where you take the you demendelize the exposure in the strata by removing the genetic effect from the strata, because of course all the low strata will tend to have more people with genetic variants related to low vitamin D. So if you stratify just by the actual measured exposure, you'll get collider bias. And then you do the MR within those 10 strata. For reasons that will be appearing in a long paper we're doing, there's very good reasons why this just simply reproduces the observational association. And the reason is, if you think of the gene environment equivalence assumption, the thing is that how the genetic variants relate, how the different sets of genetic variants relate to the exposure in different strata are different. In the very low strata, you know, people who've got low vitamin D for, you know, ill health, etc. You can have interactions between genetic variants and those other factors in the strata, where they have larger differences. Actually, there's larger differences by the instruments. The instrument doesn't have a constant effect. These are the observational estimates, and you get the observational estimate with the MR. And this was in this was a figure from that paper that I then I reproduced when I wrote into the journal to point out what you have here is an overall null, 0.99, and every one of the strata lies to the, is more protected, it shows protection, is in the protected direction. Now, this is impossible. I mean, people have heard of, the problem is people have heard of Simpson's paradox and think that this is possible. But it's not possible when MR has a causal interpretation, which is that your estimates are supposed to be causal, and you know the distribution of the numbers of outcomes. So you can look at non-collapsibility, which could generate these associations. With those things together, it is just not possible that you get an overall null and all the strata lie the other direction. I mean, if you sort of meta-analyze the strata, you'll get a reason why, how extreme the difference is. So, I wrote in, and the authors just recapitulates confounded observation findings for vitamins, which is sad. The authors then, this is their original finding, and they introduced a new method, which I will go into. I've written a couple of papers about the doubly ranked method, and their doubly ranked method didn't show the protective effect. So and we used the negative controls. This paper's published now. We used the negative control of using precisely the methods that they used, but using chromosomal sex as the outcome. So this is doing an analysis where you're looking at the effect of vitamin D on your chromosomal sex. So it's pretty obvious that that's not actually possible. But look, you get a big protective effect, against being male at the low end. So that's, of course, why you were showing beneficial effects of vitamin D at that level. But notice you see it for both methods, actually. It shows some degree of bias. And this is being older, and you get a big effect on age. Vitamin D influences how old you are. Paper's been retracted. This one is now the second paper using the same method it's had a notice of concern put up against it. This is the press release. But people are this is what really quite sad is people were publishing these papers and they put up press releases saying it's not ethical to do trials. The Mendelian randomization evidence is so strong that even though all the trials have been null, they're now saying can't do any more trials because the MR data shows it's good. Everyone should take vitamin D, and this is precisely what our genetic approach. The power of our genetic approach is that we can tell you the answer without harming people by not giving them vitamin D. Everyone should take vitamin D. And do you want to present this bit? This is a Hunt paper where the optimal BMI is 25, which is at the border of where you start becoming overweight. You didn't see it in smokers, but it was even, look at it, two and a bit fold relative risk at the low end of BMI. So you just eat more, put on weight, and you reduce your mortality if you were an ever smoker. And the conclusions were increasing BMI to the normal weight category would reduce the risk of all-cause mortality. This is strong causal claim for underweight participants, with harm of having low BMI being evident in ever smokers. So I am an ever smoker. I was quite an enthusiastic ever smoker at one time. And believe it or not, my BMI is below 25. So I've got to start eating. I've got to put weight on. If I put weight on, I'd reduce my mortality. These things are fantastic. If you see the number of calories you get per pack, a few packs of these and my mortality will be going down. Now, one problem it was that there was stratification on smoking, but smoking is influenced by BMI, so that's stratifying on a collider. That's a problem. We'd published a paper in 2018 using the method. Our main discussion was of the main effect, but we did produce a non-linear effect, even though we didn't mention it. I don't think we didn't mention it in the abstract. But once we realised that these were producing nonsense, we wrote to the journal, and they published a correction. So now, if you go into our paper, it says there's a thing saying do not ignore the paragraph on nonlinear MR effects and read this commentary where we showed the negative controls for BMI. So this, this is BMI and your chromosomal sex. So your BMI influences your chromosomal sex really quite strongly. Odds ratios of 1.2, anyway, and it influences age as well. And you can guess the answer. Can you guess the answer to this? They've now done nonlinear MR of HDL cholesterol, which is the pin-up of useful MR. That's the Observational Association UK Biobank. And the nonlinear MR just replicates the Observational Association. Huge. So this just reproduces the observational associations. And what's partly underlying all of these is the increasing GWAS size. When you do increasing GWAS size, just more and more of your variants will be variants for upstream factors. Like if you look at C-reactive protein, genetic variants related to smoking, to body mass index, to remnant cholesterol, to a whole host of factors are related to, become instruments of CRP. Because as you increase your size, you're more and more likely to pick up genetic variants that are influencing your exposure through a higher level thing. And that's the beautiful pin-up from of CRP, the CRP variant against all these confounders. If you do the GWAS, you get the GWAS score from the current CRP GWAS, you just reproduce all. So you just get it all comes back again. The genetic instrument relates to tobacco smoking, CRP causes tobacco smoking, body mass index, etc. And if you consider this in relation to gene environment equivalence, you've got cis CRP variants that, they would be equivalent to a drug which, lowered CRP, targeted CRP. Body mass index is clearly not gene environment equivalence if they work through body mass index. Smoking isn't. IL-6 certainly isn't. Anonymous, just all anonymous variants, you've no idea whether they are. Sort of aside as a multivariable MR, you need to consider the multivariable gene environment equivalence. I'll skip that. You've never heard of Gerald Wagner. So what is to be done? Journals shouldn't send out for review 2-sample MR findings, unfortunately without triangulation. Reviewers should refuse to review such. Journals shouldn't send out for review MR papers without the Strobe MR checklist. Strobe MR checklist should be reviewed by referees and the handling editor. A method should be more rigorously evaluated before implementation, including through the use of negative and positive controls. And what it should become is it should be part of triangulation of evidence. So I don't think you should just publish MR. I don't think certainly a 2-sample MR should just, they've all been done. They're all on Epigraph DB. You should think of it as a component of triangulation. Be presented with a triangulation of evidence. Reconnect with biology seems to be the mechanical application of IV analysis. Contribute to the collection of appropriate data. What we really need for looking at therapeutics are clinical cohort collections for disease progression. Then you can do disease progression MR and methods development. We applied to questions of natural selection, like paper the last couple of years was doing that with respect to lactose persistence. It's being increasingly applied to non-human life forms, which is, in non-human life forms, you don't really have this, you don't have the smoking, drinking, social class type confounding. So you're probably in a better situation. And also it's, important, because you're actually looking at things like milk production and health traits in cows. And it's really powerful. And rice production. So there's a lot of MR, and they actually call themselves Mendelian randomization. So I really love that stuff. That's useful. Be used to understand how confounding generates robust and generalizable non-causal associations. I showed this in the triangulation talk. That's an example of showing using genetics to understand confounding. Because the reason is if you have really reliable observational associations like HDL and lower risk of coronary heart disease or lower risk of sepsis mortality, underlying that there must be confounding. I mean, correlation actually does imply causation. When you get a very reliable correlation across all contexts, there must be some, there must be some very strong compounder. And if you can identify that compounder, it helps you identify causes. So using the MR principles, but to actually understand confounding generating non-causal associations. be a component of the toolbox of approaches for identifying ubiquitous exposures. I talked about that earlier, and be combined with studies of somatic mutation. And this is also what I was talking about earlier, is that, well, I didn't really get to this far, is that every single base pair in your genome will have had mutations, will have been mutated cells. So, and in 15, 20 years time, there will start being a lot of studies with vast data sets on the somatic mutations, starting with blood, but then move on to other things that are sampleable in people, and then you can use Mendelian randomization for like understanding why what are the tissue specific factors that lead to clones taking off, etc. And those are the people I would like to thank, especially very sadly Richard Gray, who gave me a lot of information about the bone transplant, the bone marrow transplant. So he's died a few weeks ago. So these are the people who did the work or gave me some information about various things. I'd especially like to thank Johan, who supported my visit within the restraints of the Norwegian socialist principles, is what he told me. So that means he's going to take me and buy me half a pint of beer. But we all know that's good for me, you know. These Norwegian socialist principles are going to keep me.
Remember, it's only the moderate drinkers who should drink.
Yeah, exactly. Yeah.
Doesn't matter for the heavy drinkers.
Maybe the Norwegian socialist principles, they want to get rid of them because they just become a burden in society. That's a 2003 paper. That's Alan Sanderson's review, which is really is the best thing available on the technicalities of MR currently, in my view. And there's a conference starting. Some people are joining remotely, I think. Or maybe Bjorn Oslo, who's already left, is doing. Sorry, I won't talk to you.
No, that's fine. Thank you. That's great. Still some people on the. Yeah.
All asleep.
No, there are nothing. They're listening all the time.
Can I ask a question?
Yeah.
So did I understand you correctly that the use of polygenic scores now, instead of single or a few more significant SNPs, is you're in a way worse off?
Yeah, I think generally having having a really good biological justification for SNPs is important. When you can have that, like for vitamin D, you don't want to take the GWAS. You use the four variants in the canonical vitamin D synthesis and metabolism pathway. You use those. For CRP, you just use the cis variant, etc. So in general, there's a couple of nice papers that will be coming out from Ellen and Sanderson, which shows that, you know, interestingly, even in situations like the CRP one, if you have multivariable MR and you put in body 2 or 3 of the upstream factors like body mass index, etc., it gets you back down to the null, close to the null. So, but that depends on you. That's sort of dependent on you knowing what these upstream confounders are.
Yeah. When you talk about multivariable MR, when you have correlated genotype scores, wouldn't it be a possibility that you increase pleiotropy when adjusting the one to the other? For instance, the childhood obesity score adjusted for the adult score, so you would in a way increase.
So multivariable MR doesn't use the score. You always use all the separate variables. So in that situation, every variant related to childhood or adulthood BMI is included as a single variant, and it takes the weighting. It takes the weighting it has from both of the 2 phenotypes. So it's not adjusting the score for the other. It's actually taking the weighting. And the 2019 paper by Alan Sanderson has a vast number of simulations to show that because everything is pre-phenotyped, which is instrumented, you don't get collider bias. So you don't collider bias in multivariable MR. So that's why it's a way of mediation, which works in a way.
There is one question from from one of the listeners here. I don't know if you can hear me. Can you put yourself on the mic?
Hi, can you hear me?
Yes.
I have a question regarding MR DAG. In one of the MR assumption regarding the direction of effect from confounder to IV, why there is like a why there is different in understanding among researchers regarding the direction of effect, because there are different MR studies which are published in different journal where they put the direction of effect. Some put the direction of effect from IV to confounder and some from confounder to IV, and even in some of the studies they have both side arrows. So
No. The DAGs which put the arrows from the genetic variant to the confounder, that is actually showing correlated plerotropy, not the exchangeability. And in fact, so I'm guilty that because I wasn't first author or last author, but the MR Egger paper included that. And I hadn't noticed. And about a couple of years after it appeared, I noticed and contacted the first senior author and wanted to write a correction to the IJE, but they didn't want to write a correction. So it's wrong. It's just that the Emma Anderson letter's right, that it is wrong showing it that way. So yeah. The arrow should go from the confounder. That's why I went at some length through the dynastic effects, for example, about how you can have an arrow down to the genetic variant, even though the genetic variant, you think you can't change your genetic variant. You can't change your genetic variant, but you can change your distribution of the genetic variant. So it's a distributional phenomenon. So there's no. It isn't that they're wrong. They're just including the ones in the Egger paper. Well, one in the Egger paper. It's wrong. Should be a correction. I should have written it on my own, but it looked a bit mean to do that.
thank you for clarifying. Thank you very much.
One more question if we have time. You talked about the jackknife procedure if you develop your or you use GWAS estimates for your MR in the same cohort or the same sample. But if you have a subsample where you identify the genes and then, for example, use data from Hunt4, but there are a larger sample from previous hunts, but it's not included in the detection sample. But of course, there are family relations. How do you think
That's interesting? My guess is if you actually simulated the effect, it would be pretty small. But you're right, that could be an issue. But everything, if you've ever done anything with, I don't think there's a handful of GWASs which you'll definitely get. If you've done MR with those phenotypes, you're doing an over precise estimate because they include UK Biobank and 23andMe. And I'm in both, and I'm included twice in those GWASs. And there will be some other people. There's more, there's more and more cases where people are being included twice, are being included twice in the study. There's a nice paper which is just on MedArchive by Jip Hamani on the effects of overlap. And of course, the bigger and bigger the studies become, the less that becomes an issue. But and in fact. Because it is definitely the most robust approach, the jackknife approach is, and it's easy to do. It's all automated. It's one to run. But in Cflang's paper, if you look at the degree of bias generated by overlap, it's really rather small. It's pretty small. So so some people might think it's too much work to do it, but it's just if you do have a method which you know is right, you should do it. But I think your basic point is that the fact that they're non-independent because of the modeling relationships.
question about the non-linear MR
Yeah.
methods. So do you mean that we cannot study non-linear effects using MR design, or should we develop better methods, or what's the?
So I think there should be said in our European Journal of Immunology paper, there should be a moratorium on using the residual and the doubly ranked and actually the polynomial, which Zoltan Kosulik put out, until there's been more reliable evidence on. You can do nonlinear MR in the way Kadori did if you've got a lot of populations with very different levels of exposure. But that's sort of, I mean, that's for many exposures, that's difficult. That's difficult to get. And there's not a single nonlinear MR paper that has actually produced any interesting findings. They're either wrong, they're interesting or wrong, like vitamin D and BMR, or they're completely trivial and wrong. So if the Gus Hamilton paper is out, so you can read that. We go through a whole series of non-linear MR papers that clearly got the wrong answer. So I can't see any reason why anyone would not to do it. Because either you just get no strong evidence of non-linearity, or you get it in the wrong direction, which is the case for the paper on lipids, which has been done, which suggests you get larger and larger effects the lower and lower people's cholesterol is, which the trials show the opposite, the exact opposite. So yeah, I would definitely not do any non-linear MR using the model, using the current methods.
Thanks a lot, George. It's so insightful to hear about this. I suggest we move to the next location. We're bringing the others also in 15-20 minutes. So thanks to those who joined online as well. I will stop now the recording and the sharing. So yeah, so thanks also to you. Jesus, we were online. Nice evening. We others, we will continue at the
Okay, yeah.